Hello everyone, welcome back to Swati's Bioscience Hub. So till now we have been discussing about the cellular aerobic respiration that includes glycolysis, pyruvate dehydrogenase complex, Krebs cycle and oxidative phosphorylations. Oxidative phosphorylation is a coupled reactions of electron transport chain and ATP synthesis or the phosphorylation of ADP. Okay, but there are some chemical substances or chemical agents which blocks the ATP synthesis process, which disrupts the ATP synthesis of oxidative phosphorylation. So these chemical substances are categorized into three types. Okay, there are three types of chemical substances which blocks this oxidative phosphorylations. They are inhibitors, uncouplers and ionophores. Okay, so today we are going to discuss about these inhibitors, uncouplers and ionophores. Okay, so let's start with the inhibitors. Okay, so let's start with the inhibitors. So oxidative phosphorylation is a reaction that combines electron transport chain and phosphorylation of ADP. Okay, so what happens? These inhibitors, these are some chemical substances which stops the electron transportation in the electron transport chain. Okay, it blocks the transfer of the electron in the complexes of electron transport chain, because electron transport chain has four complexes, complex 1 to complex 4. So what this inhibitors does, it blocks the transfer of electrons in the complexes. Okay, so how do these inhibitors work actually? Actually, these inhibitors, they bind with the components of the carriers. Okay, they bind with the components of carriers and it blocks the reduction process. Okay, if you remember, in glycolysis and Krebs cycle we get ATP along with NADH and FADH2. So this NADH, FADH2 enters into the electron transport chain and here it gets reduced and also ATP synthesis takes place. Okay, along the electron and protons, it transfers through the complexes of electron transport chain. So what happens that in these inhibitors, they block the transfer of the electron as well as the reduction process also. Okay, so there are few inhibitors that we are going to discuss. Some examples of inhibitors: it blocks the electron transfers. Inhibitor blocks the electron transfer in complexes of electron transport chain. Okay, and it also blocks reduction process. Okay, basically electron transport chain starts with the reduction of NADH and FADH2. It blocks the reduction process. So here what happens is inhibitors, there are different types of inhibitors. Okay, some examples are retinol. Okay, retinol. So this is a plant extract. Okay, it is a plant extract from Deris elliptica, that is also known as tuba root. Okay, and it acts as an insecticide. So what this retinol does, this retinol actually stops the reduction of NADH. Okay, it blocks reduction of NADH, blocks reduction of NADH. Okay, and also blocks electron transfer to ubiquinone. Okay, that takes place in complex 1. In complex 1, NADH reduces and the electron transfer to ubiquinone. So basically it disrupts the complex 1 of electron transport chain. So it blocks complex 1. Okay, disrupt complex 1. And this retinol used as an insecticide Also, it is used as a feast poison. Some tribes use it as a feast poison to paralyze the feces. Okay, so there is retinol. Second is amobarbitol. Okay, it acts just same as the retinol. Amobarbitol, that is also known as amitol. Okay, it also blocks the electron transfer in complex 1 of the ETC. It is also blocks the electron transfer in complex 1. Okay, another one is Daverol. That is also responsible to block the electron transfer in complex 1. Okay. Then number 4, there is another one, that is Persidin A. That is Persidin A. So Persidin A is a bacterial metabolite. Okay. Persidin A is a bacterial metabolite that comes from the extract of Streptomyces moverans. Streptomyces moverans. Okay, it is a extract of this bacteria, and this Persidin A is used as an insecticide. So actually what happens is Persidin A, it structurally resembles to the ubiquinone. So the electron which has to transfer from the NADH to ubiquinone, that actually transfer from NADH to Persidin A. So the electron transfer chain system at that point only it get disrupted. So it also affects complex 1. Okay. So this, then another one is antimycin. Another one is antimycin. So antimycin is also a bacterial metabolite which comes from the extract of the Streptomyces species. It acts as an insecticide, a fungicide, and also of feast poison. Okay, and this antimycin, it blocks the electron transfer from cytochrome B to cytochrome C1. If you remember, while we are studying about the complexes of electron transport chain, so cytochrome B and C1 is present in complex 3. So basically it blocks, it disrupts the complex 3 of electrotransport chain. Okay, antimycin disrupt the complex 3. So these 4 inhibitors affect the complex 1, antimycin affect the complex 3. Now move on to the next inhibitor, that is cyanide. Most of us must ever heard about this cyanide, which is known as a very toxic or very poisonous thing. Okay, so how it leads to death of the people, that we know, that cyanide. And how does this cyanide work? It is a respiratory inhibitor. Okay, so what it does, it blocks the transfer of electron from the cytochrome oxidase to the oxygen. Okay, in complex 4, the electron transfer from cytochrome oxidase to the final electron acceptor, that is oxygen. So what it does, this cyanide actually blocks that pathway. It blocks the electron transfer from cytochrome oxidase to oxygen. So what happens, ATP synthesis does not take place. Okay, so when ATP synthesis cannot take place, the body requires a certain amount of energy to carry out the life processes. So it does not have that amount of energy now. That leads to the death of the person. Okay, so this is cyanide, which affect the complex 4 of electron transport chain. Then there is carbon dioxide, that is carbon monoxide. Okay, there is carbon monoxide. So cyanide basically binds with the iron iron group that is present within the complex 4. Same, carbon monoxide also bind with the iron group that is present in the complex 4, and it also affect the complex 4. Okay, it blocks the transfer of electron from cytochrome oxidase to the oxygen molecule. So it also disrupts the complex 4. Now the next one is azide. It is also a respiratory inhibitor. that inhibits the electron transfer from cytochrome oxidase to the oxygen molecule, and this azide also binds to the iron, but here it binds to the ferric form of the iron. Okay, these two binds with the iron molecule, binds to Fe molecule, but here it binds to ferric group. Okay, ferric form of the iron molecule, and it also affects complex 4. Okay, it also affects complex 4. So these are the inhibitors which affects the electron transport chain. So retinol, amobarbital or amytal, demerol or pyridine A, these 4 affect complex 1. These 4 affect complex 1. While the antimycin affect complex 3. It blocks the electron transport from cytochrome B to cytochrome C1, and cyanide, carbon monoxide, azide, and along with hydrogen sulfide. Okay, hydrogen sulfide also, they block complex 4. So these are the inhibitors. They inhibits the electron transport from transfer in the complexes of the electron transport chain. Okay. Then next we'll discuss about the uncouplers. Okay. So as I said, the oxidative phosphorylation is a coupled reaction. Okay, the electron transport chain and the ATP synthesis or ATP phosphorylation are linked with each other. They are coupled with each other to create oxidative phosphorylation, to form oxidative phosphorylation or to form ATP. So these uncouplers are the chemical substances which break the link between the electron transport chain as well as the ATP synthesis. Okay, uncouplers are the chemical substances which breaks the link. Okay, it uncouple or break the link between electron transport chain and phosphorylation of ADP. Phosphorylation of ADP. Okay, so how these uncouplers work? Now these uncouplers, what they do, while we are studying oxidative phosphorylation, the electron transfers to the complexes of electron transport chain, and at the same time the proton also moves from the matrix to the intermembrane space. So in intermembrane space it creates proton gradient which generate the proton motive force and that drive the ATP synthesis. Okay, so here what happens, these uncouplers, they bind with the protons that are present in the intermembrane space. Okay, like in electron transport chain, protons moves from matrix to intermembrane space, and here they produce proton gradients. These proton gradients create proton motive force that drives the ATP synthesis. But what these uncouplers do, they will bind with these protons and it will bring it back to the matrix. Okay, at that moment the proton is exerted to the intermembrane space, the uncoupler will bind with it and bring it back to the matrix. That means there is no proton gradient. Okay, there is uncouplers do not let them to make proton gradient. Okay, no proton gradient. That means when proton gradient is not there, that means no proton motive force. It won't create any proton motive force. That means it can't drive ATP synthesis also. So it can do ATP synthesis, so no ATP synthesis. But while at the same stage the electron transport chain will continue, because uncouplers has nothing to do with the electron transport chain. It is just bringing that proton from itself, from the intermembrane space to the matrix. So the so here the electron transport chain will continue, but the ATP synthesis process will be disrupted. Okay, that is the mechanism of uncouplers. They will bind with the protons from the intermembrane space and bring it back to the matrix. Okay. Now let's see some examples of some of these uncouplers. Okay. So what happens when they bring it back to the matrix? So now ATP synthesis cannot take place. So that then what happens? The energy that is relieved from the electron transfer, they release as heat. Okay, they release as heat. So these kind of heat generation is known as non-severing thermogenesis. Okay, this kind of heat generation is known as non-severing thermogenesis, which is a vital process in most situations, like most biological situations, like hibernation, which is seen in newborn animals or hibernating animals. Okay, so these they are mostly seen in the brown adipose tissue. Okay, brown adipose tissue. So these newborn or hibernating animals have this brown adipose tissue, which contains a protein that is known as thermogenin. That is known as thermogenin. Okay, so this is the uncoupler here. Okay, this thermogenin will bind to the proton. Okay, bring it back to the matrix, and the energy that has to be used in ATP synthesis, now this energy will be released in the form of heat and creating more temperature for the that will be suitable for hibernation process. Okay, so thermogenin is also a uncoupler. Okay, so let's see some example of uncouplers. So basically uncouplers are classified into two types. Okay, one is chemical uncouplers, and another is physiological uncouplers. Okay, so some example of chemical uncouplers are 2,4-dinitrophenol. Okay, 2,4-dinitrophenol. Then there is one Ducomerol. Then FCCP, that is trifluoromethoxy Carbonyl Cyanide Phenyl hydrazone. Okay, difluoromethoxy. I'll provide the full form of this FCCP and another one CCCP in the description box. Okay, so here these are the chemicals. So CCCP is the Carbonyl Cyanide m-chloro phenyl hydrazone. Okay, so these are the chemical uncouplers. So physiological uncouplers, so a long chain fatty acids. Example of physiological uncoupler, long chain fatty acid. We just discussed about another one, that is thermogenin protein. Then thyroxine and calcium ion. Okay, so what is the function of uncoupler? Uncoupler will go and bind to the proton in the intermembrane space. It will bring the proton back to the matrix, so it won't create the proton gradient. So a proton motive force cannot be generated and ATP synthesis cannot take place. Okay, but electron transport chain will continue. So these are the uncouplers. Those are some are chemical uncouplers. The example are 2,4-dinitrophenol, Ducomerol, then trifluoroxymethyl, carbonyl cyanide phenyl hydrazone, then this CCCP, that is carbonyl cyanide m-chlorophenyl hydrazone. Okay, and in physiological uncoupler there is long chain fatty acid, thermogenin protein, thyroxine, calcium ion. So thermogenin are present in brown adipose tissue, which are present in the newborn animal or hibernating animal. So, and in this process it releases the energy that is driven from the electron transport in the form of heat, that is known as non-severing thermogenesis. Okay, so this is about the uncouplers. Now let's move for the ionophores. So let's go for the ionophores. So ionophores are also small lipophilic molecules. Okay, ionophores are small lipophilic molecules. It also blocks the oxidative phosphorylation, or it also blocks the ATP synthesis process. Okay, ATP synthesis, not electron transport chain. So how does this ionophores work? And this ionophores works in two ways. Okay, it works in two ways. Sometimes it makes the lipid bilayer membrane of mitochondria permeable to certain ions. Okay, it make the lipid membrane permeable, mitochondrial membrane permeable to ions, or it creates permeable channels across the membrane. Okay, so sometimes what it does, it acts as a mobile carrier. Okay, so it is intermembrane space and this is here the matrix. So how it does work as a mobile carrier? So here this ionophore will bind with the proton. Okay, it will bind with the proton and other ions, and then it will make permeable to the inner membrane. It will come inside to the matrix, and here it will release that ion what it is carrying with it. Okay, that way it is acting as a mobile carrier, and also it creates a permeable channel here. Okay, it creates permeable channel here for the movement of or the exchange of ions. Okay, in two ways this ionophores works. Okay, so now there is some example of ionophores. Okay, let's see some example of ionophores. So let's start with valinomycin. Okay, valinomycin, which is a antibiotic. Okay, valinomycin, which is an antibiotic, and it makes the cell membrane, the mitochondrial membrane, permeable to the potassium ion. Okay, it makes the cell membrane permeable to the potassium ion. That means the potassium ion now can move, can come into the matrix while the ATP synthesis is taking place. So what will happen? It will decrease the proton gradient that has to be formed, that has to be created in intermembrane space. Okay, when the potassium ion comes in, so okay, so it needs to be hydrazone ion. So when the potassium ion is coming in the intermembrane space, it will decrease the proton gradient. When it will decrease the proton gradient, it will also decrease the proton motive force. Okay, and eventually it will disrupt the ATP synthesis. Okay, so valinomycin makes the membrane permeable to the potassium ion. Next, another example is gramicidin A. Gramicidin A. So this gramicidin A will create a channel, a permeable channel for the movement of various ions. Okay, it creates a channel. Okay, permeable channel. It creates permeable channel for movement of various ions. Okay. Next, there is salinomycin. Okay, that is salinomycin. So salinomycin will make the cell membrane permeable to sodium. Okay. It will make the cell membrane permeable to sodium. That's why it is known as sodium ionophore. And it is also used as a feed for livestock, as a growth promoter. Livestock are like cattle, okay, cattle, birds, chickens, or anything. So like all the livestock, it is used for the as a feed for the livestock. Okay. Then another example of ionophore is lazuloside. Lazuloside. Okay. So lazuloside is also used as a feed for the livestock. Okay, it is also used as the feed for the livestock. Salicylomycin is creating for the sodium one. Another one is monosin. Monosin. That is also a sodium that also makes the membrane permeable to the sodium ion. Okay, that also make the membrane permeable to the sodium ion. Also used as the feed for livestock. Okay, function is also same. Now the next example is niacin. Niacin. Okay, niacin acts as a carboxyl ionophore. Okay, it acts as a carboxyl ionophore. And also it helps in the exchange of sodium and potassium. Okay, it makes the cell membrane permeable for the exchange of sodium and potassium ion. Sodium and potassium ion. Okay, so these are the ionophores. Valinomycin, which makes the cell membrane mitochondrial membrane permeable to potassium ion. Gramicid, which makes a permeable channel across the membrane for the movement of various ions. Then the salicylomycin, which acts as a sodium ionophore. Then the lazuloside, which acts as magnesium. It acts as a magnesium and calcium ionophore. Okay, it acts a magnesium and calcium ionophore and also used as a feed for livestock. Then the manosin, which also acts as a sodium ionophore. Then the niacin, which has a carboxyl ionophore, and also it helps in the exchange of sodium and potassium. Okay, so some of these ionophores are used as feed for livestock, as a growth promoters, and also used in the veterinary medicine for a disease called coccidiosis. Okay, a disease called coccidiosis. In livestock only, it is used as a veterinary medicine for disease called coccidiosis. And in research, it is used to study the cell biology, the cellular physiology, and membrane transport. Okay, it is used to study the membrane transport or the cellular physiology. Okay, and some of these ionophores like valinomycin, salicylomycin, lazuloside, manosin, then the niacin, these are used as a potential anti-cancer drugs. Okay, they are used as means they are investigated to be used as a potential anti-cancer drugs. Okay, so these are the chemical substances which blocks the ATP synthesis process. They are known as inhibitor, uncoupler, and ionophore. So inhibitors, they actually inhibit the electron transfers in the electron transport chain, in the complexes of electron transport chain, and in uncouplers, they uncouple the electron transport chain with the ATP synthesis, and it disrupt the ATP synthesis process. And same as ionophore, uncouplers bind with the protons in the intermembrane space, bring it back to the matrix, and it disrupt the ATP synthesis by disrupting the proton gradient or proton motive force. Same with the ionophores, it also let other ions enter into the membrane, so that's how it decrease the proton gradient and it disrupt the ATP synthesis. So this is about ionophores, uncouplers and the inhibitors of oxidative phosphorylation about today's discussions. So if you have any doubts regarding this, please comment in the comment box. If you like my video, please like, share and subscribe to my channel. Thank you so much.